Welcome to this new video about the organization of cells. This time we will see the structure of the cell membranes, and I would like you to keep in mind that if we do a focus on the plasma membrane, most of what we will see today applies also to all the membranes that are found in cells, including the envelope of the nucleus or of the mitochondria. So keep in mind that this is not only the plasma membrane that we see here. So first of all, what are the functions of membranes? It is their first job is to form compartments, so to separate compartments that are made of an aqueous solution, including all the solutes that are dissolved in the watery environment. They will separate the exterior and the interior of the cell, as well as the exterior and the interior of all the compartments like the lysosomes or the Golgi network. They will also provide a scaffold for biochemical activities. It means that a lot of the membranes will contain proteins that can act as enzymes in order to catalyze biochemical reactions. They act as a selectively permeable barrier, so they will not allow everything in, but they are still permeable to several solutes. So this is why we call them selectively permeable barriers. It is because not everything will enter, but some things will enter. They are also involved in the transport of solutes. So it is possible that solutes will be able to enter, but they will need to be transported by the membrane. So the membrane will have specific features that will move the solutes in and out of the cell or the compartment. They will be responsible for the response to external signals, so it means that they will carry receptors that will be able to sense external signals. And they are responsible for the trafficking intracellularly, so they will form little vesicles that will contain various solutes, proteins, and stuff that need to be transported at various places of the cell. So these vesicles made of membranes will be important for the trafficking of these solutes. And finally, they will provide a scaffold to transduce energy. It means that the membrane will form a barrier that can act to prevent the transport of solute, but also to store the force, the mechanical force of the solutes that are contained in one place. So they will be responsible to transform this form of energy into a different form of energy. Let's now have a look at the organization of the membrane. It is a structure that has, as we say, a fluid mosaic model. So, in fact, it is to say that the membrane is a structure that is far from being fixed and inactive. In fact, the organization under a lipid framework will confer a fluidity, and this will allow the membrane to be in constant motion in every direction, and all the structures that are inserted or associated to the membrane will also be in constant motion. So, how is it organized exactly? First of all, there is a frame of lipids, and this frame forms a lipid bilayer. So, you have two layers of phospholipids, and it also contains several molecules of cholesterol um, inserted between the phospholipid molecules. So it is all the grayish heads and tails that you see here on this picture, and cholesterol molecules are represented in yellow. So this forms the frame in which other molecules will be inserted or to which they will be associated. What are these molecules exactly? First of all, proteins. A lot of proteins are inserted directly into the membrane. They're represented in pink here on this picture. Various molecules then totally inserted into the lipid bilayer, or some of them can also be simply associated at the periphery of the membrane, so on the cytoplasmic side or on the extracellular side. You can both sides, you can have proteins that are associated to the membrane. Finally, you will also find sugar molecules on the membrane, exclusively located on the external face of the plasma membrane, never into the cell, or at least never into the cytoplasmic side of the cell. So they contain little polysaccharides. We also call them oligosaccharides. They're made of around 10 to 50 sugar molecules that are associated by glycosidic bonds, and they will be found either on top of a protein, so in this case we call them glycoproteins, or directly attached to a lipid, and in this case they are called glycolipids. Together, these carbohydrates will form what we call the glycocalyx, which is the jelly-like substance that surrounds every cell, and it is just because of the properties of sugars that can attract a lot of water molecule, so they will form a bit like a jam around all the cell. Now we will review all the elements one by one, so let's have a closer look at the lipid bilayer that forms the frame of the membrane. The most important lipid forming the membrane is called a phospholipid. It is not the only one found in membranes, but it's the only one we will see in this class. It is a molecule that is called amphipathic because it is a molecule containing a hydrophilic side and a hydrophobic side. The general organization of the molecule is always the same. It has a polar head group that is attached by the mean of a phosphate to a molecule of glycerol. This attachment by the mean of a phosphate group confers the name phospholipid. If you had no phosphates, it wouldn't be called a phospholipid, so it is important to remember because there are also membrane lipids that don't have a phosphate and that are not called phospholipids. The fatty acid tails, two of them, will be attached to the polar head groups, again by the mean of the glycerol molecule that forms a kind of scaffold between the fatty acids and the polar group. A bit more precisely, when you look at phospholipids, there is always a backbone. So in the previous slide we saw the glycerol as a backbone. It could also be another type of molecule called the sphingosine. You always have fatty acids attached to the backbone. In the case of glycerol, it is called a glycerophospholipid and you have two fatty acids. In the case of a sphingosine backbone, in contrast, you have only one fatty acid because the sphingosine itself has a very long carbohydrate chain that is strongly hydrophobic, so it acts a bit like a fatty acid itself. And there is always a phosphate group in both the glycerol and the sphingosine backbone phospholipids. Finally, it has a polar head group. The polar head is not always the same. There are 6 possible different polar heads called choline, ethanolamine, serine, inositol, glycerol, or bisglycerol. And depending on the type of polar head, the phospholipid will have a different name. For instance, a phospholipid containing choline will be called phosphatidylcholine. If it is a phospholipid containing ethanolamine, it will be called phosphatidyl-ethanolamine. So due to the chemical nature and properties of phospholipids, they will form stable structures when they will be put in contact with water or with aqueous environments. One of these structures is called a mycel. It is the simplest structure, and it forms a sort of sphere where all the tails of the phospholipids will merge in the center of the sphere, while the hydrophilic heads will cover the sphere, creating a kind of shell, and will remain in contact with water. Mycel formation is most often seen with fatty acids, simple fatty acids, while when you have phospholipids, because they're a bit bigger, they will more spontaneously form bilayers, like the structures shown here. In fact, a bilayer forms where you have the hydrophilic heads will be in contact with the watery environment. So there will be one top and one bottom layer of hydrophilic heads, and the hydrophobic tails will be in the center, so tail to tail, and will create a thick layer of hydrophobic molecules, in fact hydrophobic groups, and to which water will be repelled. One problem here comes from the fact that water molecules could eventually get in contact with the hydrophobic tails from the sides of the bilayer. So this is why what you will see most often in a watery environment or in an aqueous solution is again a sphere, but a sphere containing two layers where you have watery environment in the middle, like in the center of the sphere, and a watery environment outside of the sphere, so in periphery. And both the internal and outermost layers will be covered by the hydrophilic heads, while the hydrophobic tails of the two layers of phospholipids will again create the thick bilayer, but this bilayer will form a sphere so that no hydrophobic tail can be in contact with water molecules. So a liposome, so this spherical structure of a lipid bilayer with the two, in fact, with the watery environment separated in two parts, one in the middle and one in periphery, will occur very spontaneously when you put phospholipids in contact with water. You can also consider that all the vesicles and cells themselves are kind of big liposomes because they are made, their membrane is made by a bilayer like this that forms a spherical structure. Now, when membranes are formed, it is important to understand that the nature of the fatty acids found in the tails of the phospholipids will influence the tightness, the packing, in fact, how fluid the membrane will be, and also how thick it will be. You remember that every phospholipid is made of one polar head and two fatty acid tails. So these fatty acids, if they are very long, for example, it will make the membrane thicker. If they are more saturated, they will create a membrane that is more compact and less fluid. If they contain lots of unsaturations, they will in contrast create a membrane that is more fluid and less compact. Another molecule is important in controlling the level of fluidity of the membrane, and it is the cholesterol molecule. The cholesterol molecule is another type of lipid. It has a very different structure if you compare it with the phospholipids. However, it still has two properties that are the same: one hydrophobic hydrocarbon tail and one hydrophilic head group, even if it is very small. And this structure allows the cholesterol molecule to be inserted between the fatty acid tails of the phospholipids. And this molecule, because of its rigid planar structure, will create a kind of perturbation in the membrane. So, because you remember we said that the phospholipids are in constant motion, they will often bump on the cholesterol molecules. So a rule of thumb is that if you have a few cholesterol molecules, like between 10 and 30%, it will increase the fluidity because the hydrophobic tails will bump on it and it will increase the level of motion. In contrast, if you have more than 30% of cholesterol molecules in a membrane, then it will increase the rigidity, so decrease the fluidity, because it will pack too much and it will prevent motion in this case. Now, another important molecule found in membranes is, in fact, are proteins. We said previously that the membrane has a frame made of phospholipids, but into this frame are inserted or associated a lot of proteins in the fluid mosaic model. So we'll see a little bit of these membrane proteins. And the first type of membrane protein I want to cover with you is the integral membrane proteins. So these proteins are called like that because they cross the membrane entirely. So you have a part of the protein that is found in the extracellular fluid. You have one transmembrane domain, so a domain of the protein that crosses the membrane completely, and another part of the protein is found in the cytoplasm. So these membranes are called integral because they are both in the outer face, in the middle, and in the inner face of the membrane. They can contain more than one transmembrane domain. If they have just one, it looks like the protein at your left. If it has more than one, it will make loops in the cytoplasm or in the extracellular fluid so that you can have more than one transmembrane domain. Sometimes you can have several transmembrane domains that form, for example, a channel or an opening in the membrane. Now, you can also have proteins that are not integrated to the membrane, so we won't call them integral membrane, but they are in contrast found in on the outer face or on the inner face of the membrane, and because they're not inserted into the membrane, they're just associated to one face, they're called peripheral membrane. So the big difference with these proteins, so these are the green proteins that you can see here, is that they don't have a transmembrane domain. And how can they remain attached to the membrane? It is by two possible ways. You can have them attached to another protein that is itself an integral protein. So the peripheral membrane protein will be attached to either the inner face or the extracellular domain of the integral membrane protein. Or they can also have a little lipidic addition, like a fatty acid, that is added at the end of the protein and that anchors it into the membrane by the means of this hydrophobic tail. What are the functions of the membrane proteins now? So several functions can be achieved by proteins that are inserted in the membrane. First of all, transporters. Transporters can include channels that will create an opening that ions or small molecules will be able to take in order to move from one side to the other side. You also have carriers that are molecules that will transport ions or molecules from one side to another, but by making a direct interaction with the transported substance. Proteins can also serve as receptors, so some of the proteins will be able to sense extracellular substances and to send intracellular signals in the cytoplasm of the cell. You can also have enzymes that are inserted in the membrane and catalyze chemical reaction either in the extracellular fluid or in the intracellular fluid. So, for example, you can think of membrane proteins that will be able to digest the extracellular matrix to be able to move and crawl to another place in the tissue. And finally, proteins can serve as anchors so they can be able to interact with other proteins or other molecules, either extracellularly or intracellularly, so that the compartment created by the membrane in question will be attached to another part. So, for example, an intracellular anchor can attach to the cytoskeletal filaments and maintain the structure of the membrane, or extracellular anchors can attach with extracellular matrix molecules or to other cells' proteins, so that it attaches the cell to a substrate or to another cell. Now, more specifically to the plasma membrane or lipid rafts. So, lipid rafts are specific areas of the membrane where you have more proteins, where you have an increased thickness and rigidity, so that you increase the potential of interactions with the extracellular environment. So on the top picture here, you can see the lipid rafts in red, and they are in a membrane that is shown as the darker spots. In fact, all the black reddish is the plasma membrane, and the rafts are the bright red structures. And how are they made exactly? So they're not floating at the surface of the membrane, but they are part of the membrane itself. They're just made of phospholipids and sphingolipids. They have longer tails in their fatty acids. In addition, their fatty acids are more saturated, so the tails are more straight and less kinked. Therefore, it increases their thickness, their height. They are also areas where you have an increased concentration of cholesterol molecules, represented in yellow here, so that you will pack more and tighten the lipid raft. They are also areas where you have an increased concentration of sugar additions at the surface of the phospholipids. So, in fact, the molecules that we call glycolipids will be found more frequently, and they are meant to interact with extracellular environment, extracellular substances, and other cells. And finally, you will have lots of proteins that are added directly at the surface of the phospholipids or on the glycolipids when they serve for the recognition and adhesion to other cells or other extracellular substances. Speaking of the glycolipids and sugary additions to the plasma membrane, I want to highlight the fact that membrane sugars or membrane oligosaccharides will be extremely important for the recognition and adhesion to other cells and to extracellular substances. So, as we said previously, these carbohydrate chains are made of simple sugars added to a lipid or to a protein, and usually you have between 5 and 10 and sometimes a bit more sugars that will be added to the protein or to the lipid, and will form a little chain that will be able to attract water and form the glycocalyx, or this jelly-like substance that is found at the surface of every cell. They can be, as we said, added to proteins, and in this case they're called glycoproteins, or they can be added to the phospholipids, and in this case they're called glycolipids. Their main function is a recognition and adhesion to other cells. So this closes the video about the structure of the plasma membrane.